Hello, everyone. We are the members of Group 10, the MCAT Warriors, and today we'll be presenting Inhibition and Enzyme Kinetics under Content Category 1A. So, enzyme kinetics for dummies. So, what are enzymes anyway? Enzymes are proteins that help speed up the chemical reactions in our bodies. So, how do enzymes work? Enzymes perform the critical task of lowering a reaction's activation energy. That is the amount of energy that must be put in for the reaction to begin, shown here in the free energy diagram on the left. Enzymes work by binding to reactant molecules, also known as substrates, and holding them in the enzyme's active site in such a way that the chemical bond breaking and bond forming processes take place more readily. So in the diagram, we start with our reactants. The black curve shows the activation energy needed for the reaction in the absence of the enzyme, and the red curve shows the activation energy of the reaction in the presence of an enzyme. One thing to pay attention to is that the enzyme does not change the change in free energy of a reaction, which is also shown in the diagram. There is no difference between the energy of the reactants and the products between the catalyzed and uncatalyzed reactions. Instead, the enzyme lowers the energy of the transition state. Another important thing to note about enzymes is that they can be regulated by other molecules that can increase or reduce their activities. The molecules are called either activators or inhibitors and bind to the enzyme either at the active site or at another site on the enzyme, also known as the allosteric site. So now we will move on to the breakdown of simple steady state kinetics. The Michaelis-Menten equation relates the reaction velocity to the substrate concentration for a system where the substrate S binds reversibly to the enzyme to form an enzyme substrate complex, which then reacts irreversibly to generate a product and to generate the free enzyme. In the equation, V naught, which we call Victor velocity, is the initial velocity of the reaction. And that V naught is equal to V max, which here we call speedy Gonzalez, and represents the maximum velocity achieved by the system. Km, the Michaelis constant, or can half of the way, is the substrate concentration required to reach 50% of the Vmax. And S, or substrate Sally, is the concentration of the substrate. One important thing to note that this equation assumes that during the reaction, the concentration of the enzyme substrate complex remains constant and is lower than the concentrations of unbound substrate. These conditions are known as steady state. On the right, we have the Lineweaver-Burke equation, a double reciprocal equation, which is in the form of the equation of a line and can be used graphically to determine the values of Km and Vmax. In the resulting graph of this equation, the y-intercept is equivalent to the inverse of Vmax, and the x-intercept on the graph represents -1 over Km. The graph also gives a quick visual impression of the different forms of enzyme inhibition.
Here, we will briefly touch upon the different types of inhibition shifts you can encounter in enzyme kinetics. We have competitive, uncompetitive, and mixed, also known as non-competitive and irreversible inhibition. It is important for you to note the red flags of each type of inhibition, such as where is the inhibitor binding, the active or allosteric site, and how does this binding in turn affect the Km or Vmax? These inhibition shifts can further be categorized into reversible and irreversible types. Reversible inhibition is characterized by the non-covalent binding of the inhibitor to the enzyme. This type of bond occurs through hydrogen bonds, hydrophobic interactions, and ionic bonds. These type of bonds are weak, and they allow for the enzyme inhibitor complex to easily dissociate, thus restoring enzymatic activity. This includes competitive, non-competitive / mixed, and uncompetitive inhibition. Next, we have irreversible inhibition, where the inhibitor binds the enzyme through a covalent interaction, which can modify the amino acid by reactive functional groups. These types of bonds are strong, so this allows for the slow dissociation of the inhibitor from the enzyme, and the enzyme will take 5 to restore its action. An example of irreversible inhibition is a suicide inhibitor, such as penicillin.
And now to get into each of those inhibition ships, we'll start with the competitor. And the competitor is also known as a competitive inhibitor. And it gets its name because this inhibitor competes with the substrate for the enzyme's active site. In this competition, if the inhibitor wins, its prize is a higher Km value. This is because once the inhibitor binds to the enzyme, it decreases the enzyme's affinity for the substrate. And if the enzyme's affinity for the substrate is decreased, then there will be an increase in the Km value. For the competitive inhibition, the Vmax remains unchanged, which you can see both the Vmax and Km in these two graphs, which are the Michaelis-Menten plot, which is A, and the Lineweaver-Burke plot. And here you can see how a competitive inhibitor is represented in each of these plots in comparison to a reaction that is not inhibited. So like I said before, there will be a higher Km value, which you can see in the Michaelis-Menten plot, which is in red, and also on the x-axis of the Lineweaver-Burke plot. Then again, you can see that the Vmax is the same in both the Michaelis-Menten plot and the y-axis of the Lineweaver-Burke plot. In addition to the competitor, this type of inhibition can be overcome by an increase in substrate concentration. This is because if there's more substrate, it's more likely for the substrate to bind to the enzyme than for the inhibitor to bind. A real-life application of a competitive inhibitor is methotrexate, which is an antineoplastic drug used for chemotherapy. An antineoplastic drug acts in preventing, inhibiting, and halting the development of a neoplasm or a tumor. And so methotrexate binds to dihydrofolate reductase, which is an enzyme responsible for the synthesis of certain amino acids. Once methotrexate binds to dihydrofolate reductase, it interferes with DNA synthesis and blocks cell division and rapidly dividing cancer cells. And because this is competitive inhibition, it is reversible. Next, we have the latecomer, or also known as the uncompetitive inhibitor, and it gets its name because this type of inhibitor binds to the ES complex, which is the enzyme substrate complex. So it binds allosterically, and once it binds, it decreases the Km value and also decreases the Vmax. The reason why it decreases the Km value is because once the inhibitor binds the ES complex, it becomes an ESI complex, which decreases the concentration of ES complex. So to compensate for that, the reaction increases the enzyme's affinity for the substrate, and with a higher affinity for the substrate, that decreases the Km value. As far as Vmax is concerned, once the inhibitor binds to the ES complex, the enzyme is disabled from creating product, which is why the Vmax decreases. An example of an uncompetitive inhibitor is camptothecin, which is another anti-cancer drug. And this drug binds to topoisomerase 1, which is a type of enzyme that's responsible for overwinding and underwinding DNA. And you've seen that in its ability to remove supercoils during replication and transcription. and also disentangling DNA during mitosis. And it also has the ability to re-anneal DNA. So once camptothecin binds to topoisomerase 1, it inhibits its ability to religate DNA.
Okay, so now we have the free spirit, also known as the non-competitive inhibitor, which is also known as a mixed inhibitor. The most important note about this inhibitor is that it binds allosterically to an enzyme and combined either before a substrate binds to the enzyme or after a substrate binds. This type of inhibition does not affect the Km, but does decrease the Vmax, as shown in both the Michaelis-Menten and Lineweaver-Burke plots.
Here we present the party pooper, also known as the irreversible inhibitor. This inhibitor covalently binds the enzyme, thus inactivating it altogether. This action does not affect Km, but decreases the Vmax. The Michaelis-Menten plot for an irreversible inhibitor resembles that of a non-competitive inhibitor. The only way we can distinguish between the two is by viewing the Lineweaver-Burke graph. In this case, the enzyme will be inactive until all of the inhibitor is used up. A real-world example of this type of inhibitor is cyanide. So cyanide is a poison that prevents ATP production via aerobic respiration. For all of my electron transport chain fanatics, cyanide will bond to the allosteric site on the cytochrome oxidase, thus blocking the activity of complex 4. Just to reiterate, this process is irreversible.
And now we have come to the finale of our presentation. So today we've learned a lot about enzyme kinetics, specifically in relation to inhibition. Starting off, we have that the enzyme is a protein catalyst that decreases the activation energy by stabilizing the transition state. And these enzymes can be affected by inhibitors and activators that will either decrease its ability to function or increase its ability to function. And these inhibitors and activators can bind to the active site where the substrate will also bind or can also bind, and also a allosteric site. Then for inhibition, we have two different types, ones that are reversible and ones that are irreversible. As far as the reversible ones, there are three, which is competitive, non-competitive, and uncompetitive. And a great way to distinguish between these three is to focus on particularly where the enzyme binds, whether it's the active site or the allosteric site, when the enzyme bonds, which is whether it's after enzyme has binded with the substrate or before the enzyme has binded with the substrate, and also how that binding affects the Km value and the Vmax. You can also use the graphs of a Michaelis-Menten plot or a Lineweaver-Burke plot to help you distinguish between the different types of reversible inhibition as well. Finally, we have for you a mental cheat sheet to help you kind of put together and remember the different types of inhibition, specifically the reversible inhibitions. So these are three different ways to do so. Some are graphical, some are written, and you can just little ways. So you can pick out of these three which one works best for you to help you remember. And that concludes our presentation.